then can we double check with the slides?
Do you want to go back to presenter mode and just like go between the slides and double check that YouTube is also doing that?
Yeah.
And also like get somebody to listen to the lecture to see if there's actually any noise.
Yep.
Okay.
And then he likes to have the laser pointer, so if you go back to the presenter view, and then that pen thing, click on it, and then go to laser pointer.
Last thing, can you try to go on Facebook? Because I think Andrew won't work. I just want to see.
Like just go on Facebook? Do you want to log in? Because I don't think I'm that. Am I?
No, we're not in the new group.
No, actually, just to see in the group. I wanna check the link because sometimes they're weird. Oh my god, I don't even remember my password. No, I don't care about that. I don't care about that.
Are you having issues with
Nope,
connection?
it's all good.
Just double check.
We're just triple checking everything. It's actually working really well.
It's like relearning the entire thing to remember all this stuff.
Yeah. So we had problems before where you click it and then it's a window within a window. And it's weird.
Sorry, as you're coming down, there's coffee and some snacks for you on the
This is what happens.
If you need any hard copies of today's lectures, I have a few copies for you at the front.
What if they click with an yeah. So they just this is what happens.
If you need any hard copies of today's lectures, I have a few copies for you.
Just click on it to get to the live stream. Great. Okay, so when he finishes, go back to OBS. It was working. I don't know if it's... Can't we just unlink the screen over here and then see what the sound is? Like if you go to YouTube, the community. Working. I don't know if it's...
Hello? Do you hear anything?
I mean, you would hear it because he doesn't have headphones. You just did. It's okay.
Hello? Do you hear anything?
I mean, you would hear it because he doesn't have headphones. It's okay.
Hello? Do you hear anything? 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 Hello?
Hello, hello. 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 Hello, hello Hello, hello. 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 Hello, hello.
but we do have alternate recording
Let's
links
wait for
for
a
you
second.
as well, and we want to make those available for every single class that you have.
And this one is gone, which is good. It was because YouTube was open.
You
Can
think
you
there's
hear
sound?
Excellent.
Okay, awesome. Thank you. Okay, and I don't hear anything through the headphones, which is even better. So we are going to have occasional workshops, guest lectures. The first issue that we're going to be looking at is the gut microbiome, as we've outlined for you here. There's almost like a little mini brain that lives in your gut. We have the same number of neurotransmitters in the gut that we do in the brain. In fact, most of the neurotransmitters, before we found them in the brain, were actually identified first in these peripheral systems, and we'll be going over those. The gut microbiome is a really hot topic right now in neurobiology, and we're going to be looking and examining this in a few lectures. So I don't really have anything to disclose to you. I just really want you to do well in this course, and that's my principal aim, is to really get you interested in neuroscience, really get you interested in neurobiology. I want one of you, maybe, in this class to maybe think about possibly winning the Nobel Prize, because I'd like to be up there on stage with you when you get your Nobel Prize, and you can always say, I credit this all to those stunning lectures that we used to have way back in the day at the University of Toronto. So I do have an ulterior motive, and I want to help you get to those different types of places. I'm going to talk to you about these recognized study groups in a moment. I don't set those up. I'm not involved in it. If you guys want to do recognized study groups and you want to have me come in and help you, I'm happy to do so, but I have no formal connections with those. Again, just for the sake of time, I'm not going to go through this. We are going to have some polling in class. That's not your participation. It's more for fun than anything else. But I'm hoping that you're in neurobiology and in neuroscience because you want to be here, that you really like neuroscience, or you're really fascinated by different aspects of neuroscience. And I'm hoping that many of you are enrolled in a research project course, because that's also really important for me. If you don't have a research opportunity by the time you finish the University of Toronto, in many ways I feel like I've failed you as a prof as well. And some of you are going to be asking yourselves, like, how do I even get involved in research? And that is something that we want to teach you outside of the class, but we feel that that's really important for you as well. So if you're thinking, I'm not in a research project right now, but I really, really would like to be, then please let me help you with that. The only other thing I'm going to ask you to do, and this was a paper I was reading right before I came to class, which was actually fascinating because it actually suggests that these polyunsaturated fatty acids, there was a huge paper that came out last year, and it said that if you have a psychotic disorder, that you can be treated simply by changing your diet. And because we're interested in diet, the gut microbiome and the effect on the brain, this was a really important paper. But this recent paper actually overthrows all of that data. It actually suggests that if you change your diet and take more polyunsaturated fatty acids in your diet, that it doesn't help you with bipolar disorder, schizophrenia, any type of traumatic stress disorder. So diet now doesn't seem to be as important as it was a year ago. But I want you to get involved in these controversies, to learn how to read papers, to go and spend time reading these papers because they're really interesting. So I want this course to be really all about you. I am going to tell you a little bit about my own sort of personal journey in neuroscience throughout this course. I really want to light up your brain in various ways. To be honest, some of these topics that we're going to be covering, for some of you it's going to be really easy because you have the background. For some of you it'll be really challenging because you haven't had that background. And so sometimes you're going to be a little bit more anxious, and sometimes you're going to be a little bit more stressed out, and at other times you'll feel like it's stuff that you've already learned. But I want you to actually be challenged to learn and to develop in this course. I also want you to think about yourselves as great students, not just good students, but I want you to be great students in this class. And I want you to, again, come and see me if there are any type of thing that I can help you with. So if you have an accessibility issue that you'd like to talk to me about in private, please come and see me. I'm happy to help. If there are things that you don't understand in class and you've gone and researched it yourself, or you don't have time to research it yourself, but you're really curious about it, then please come and see me as well. It's about the learning. and hopefully about your development as a neurobiologist. Do you guys have any questions so far, or any suggestions or things that you'd like to try to do in this course? Nothing so far? No? You're thinking about it, right? Okay. So my office is over in Wetmore Hall. It's in the Human Biology Program office. You're welcome to come and see me by appointment. I have also set up some office hours for you. If I'm not available during the times that you're available, please make an appointment. I'll try my very best to make sure that I can answer all the questions that you might have. If you have an urgent administrative issue, please try to use my uToronto. So you've missed an assignment, you're really worried about it, or you have other issues going on where you're going to miss a block of classes, my uToronto email is probably the best one for you to try. So I'm going to go through some of the expectations, how you're going to be evaluated, who's going to be evaluating you on these different types of things, why we're doing the types of projects that we are, and where we're going to go in this particular course. So again, my primary thing, and the reason why I really want to teach this course, is to get you excited by neurobiology and all the interesting fields that are occurring now in neurobiology. I don't know if you guys read a lot of papers over your break. Did you? You can be honest. This is, again, things that we say here in 300 stay in 300, right? How many of you read at least one paper? That's okay. 2 papers? 3 papers? Oh, we're going to do last man standing. Okay. 5, 6, 7? Okay, 7. All right, we have a good sort of goal to look at. I want you to realize that over the course of the holidays, our understanding of neurobiology actually changed fundamentally. So I'm going to talk to you about mental health. I'm going to talk to you about bipolar disorder. I'm going to talk to you about depression. But over the course of December and January, our understanding of depression and bipolar disorder actually changed fundamentally. We didn't know the things that we knew in December prior to that. And so your class is the first class that's actually going to be examining the things that we now know about how we identify and how we might have biomarkers for things like depression and bipolar disorder. So we want you to start reading papers, to get excited by what's changing right now in neurobiology. I want you to also stay connected in this class. You're going to be seeing each other off and on over the next couple of years in other kinds of neuroscience and cell biology, psychology courses, and I want you to stay connected in this class. The best thing I saw all morning outside of people drinking coffee and tea was that people were happy to see their colleagues and their friends coming into the same class. I love that. I love seeing that you're already well connected, and I'm hoping that you'll continue to stay connected. So the other thing that I want to suggest, and it's just a suggestion, some of you are going to have your own sort of study methods and study techniques, and that's great. I'm not going to try to influence that. Again, let me ask you, these are 3 different cell types. Can you identify any of these cell types? You don't have to worry about it on a term test or anything else. I just want to know if you can identify any of these. Any of these. So I think I heard astrocytes. So which one of these are astrocytes? I'll tell you that one of them is an astrocyte, or a picture of an astrocyte. Which one? Top, middle, or bottom? Middle? Is that a guess? It's an educated guess. That is an excellent educated guess. How about the cell that's up there at the top? Any ideas? So this would be another example of a cell type that we find in the brain. It's very rare, but we do find them. Any ideas what that might be? So it's a microglia, absolutely. And what's the thing there at the bottom? So this is a neuron. So you have a choice in this course to be like a microglia. A microglia is kind of it's an immune cell, so it's not really part of the brain. It kind of infiltrates the brain. It hangs out once in a while in the brain. So you might come to class once in a while. You might kind of engage in a couple of topics, but you're not really part of the class. You might be an astrocyte, so you connect with lots of different aspects of the course, but you're not firing on all cylinders. You just modulate activity. And you could be like that type of student. You could be like an astrocyte. I'm just happy to be learning about different things and to connect with different people in the class. Or you could be this neuron, which is what I want you to be. So you're connected to all of these different cell types, and you're firing on all cylinders. You're generating those action potentials, and you're learning and modifying synapses, and you're modifying your own knowledge by being engaged and connected with other people. So I'm hoping that you're not just going to survive in this class. That's not my goal. My goal is to help you thrive and do really well in this class. So I'm hoping that you'll figure out ways that I can help you, and you can indicate all those different ways to let me help you fit into this class in the way that you feel the most comfortable. So in this course, there is no textbook. I'm not a big fan of textbooks for a variety of reasons. This is going to be lecture-based. I'm primarily going to be looking at a number of different primary papers, and I'll show you what I mean by that in the second half of the course. I'm not going to try to trick you on anything. The goal is really to help you learn and to be a better neurobiologist, not to try to lower the class average, not to try to play around with the class average. It's to really get you to learn things that are important as neurobiologists. So there's nothing from a diagram. If I don't mention it in a lot of detail, it's not something that you should be worried about. I'm going to ask you eventually to start posting things on the discussion post, and you'll be able to practice your short answers, and you'll see what I mean by that a little bit later on. So I'm hoping that all of these different activities give you some ideas as to what sorts of things we're going to try to do. I'm also going to highly suggest to all of you that we have these review sessions before tests and exams and assignments, and I ask the TAs to come in and help a little bit with that. And I want also to talk about topics that are relevant for you as neurobiology students. I don't want to talk about the action potential, to be quite honest. I don't want to talk about the resting membrane potential. Do you guys love that stuff? If you love that stuff, go take CJH 332. We'll talk about it there. If that's not really what you're interested in and you want to learn about biology and behavior, this is the course that I'd like you to be thinking about. So we're going to talk about modern imaging methods, some of the genetic and molecular techniques. We're going to talk about the microbiome, the effect of exercise on the microbiome, the effect of exercise on the brain, as well as the emerging field of epigenetics and human behavior, and how all of these different aspects come together. They're all interrelated, and we're going to try to see how they're all interrelated in this course. And we're going to see that all of these things now, it may seem like a radical idea, and up until 5 years ago, it was a radical idea that what you eat and the things that you have growing in your gut will actually determine whether or not you develop Parkinson's disease. Do you like that idea? That you can get Parkinson's disease simply by changing your gut microbiome. Is that a really cool idea? Is it a really interesting idea? Is it an idea that you've heard before? No? So I'm hoping to convince you that these emerging fields are ones that are really important, both for mental health as well as in neurodegeneration. I'm hoping also that neurobiology is going to have a personal impact on you. As a neurobiology student in undergrad, I took physiology courses in my undergrad, was here at the University of Toronto, and I did a physiology undergrad. And to be honest, my concentration was in neurophysiology, but I really didn't do well in neurophysiology. I got some of the worst marks that you could imagine, C minuses, Cs. I really didn't understand like what my profs were talking about, and I really didn't like neurobiology at all. And it wasn't until much, much later that I had an appreciation for what neurophysiology and neurobiology meant. So I want it to be personal, and I want you to understand that neurobiology should be impacting you on a personal level in many different ways. So we are going to talk about some of these genetic, epigenetic techniques, and again, some of you will be really comfortable with that because you have a strong cell molecular background, and some of you might be more comfortable when we talk about things like bipolar disorder or depression or generalized anxiety or other types of mental health conditions. And then we're also going to try as much as we can to talk about some of the emerging roles that neurobiologists have, both in terms of ethics, the policy, and the development of different types of methods. So we will talk about various mental health disorders, traumatic brain injury, as well as Alzheimer's disease and Parkinson's along the way, in keeping with these themes. Any questions so far? Any suggestions so far? No, too early in the semester and too early in the morning, which is totally understandable. So again, I want to unpack the course really briefly for you. And again, primarily in HMB 300, we're going to be looking at lectures, and we're going to provide you with as many resources that you're comfortable with, whether it is a YouTube stream or whether it's an audio recording. We want you to make sure that you have things that are going to make you feel comfortable in concentrating only on the lectures. There is sort of a writing assignment, and it's packaged together as a multimedia assignment. And part of that is the way that students are now asked to communicate is changing as well. And I'll get to this in a few moments. By the way, if you ever feel like this, and again, I want you to take this very seriously, if you ever feel like you're drowning in this course, if you ever feel like there is a lot happening to you at the same time, I want you to feel comfortable coming to see me. You can do so in private. You can talk to one of the TAs who I'll introduce in a few minutes, but I want you not to feel like you're drowning in this course. Often in undergrad when I was here, I felt completely lost, and I felt like I was here by myself, even though there were tons of other people around. And I had a lot of smart people in terms of my profs, but I really felt like I was completely lost the vast majority of the time. And I felt like I was drowning, to be quite honest. And it wasn't until I got into grad school that I learned to speak up and to ask for help. And if you ever feel like you're drowning, and if you ever feel like you need help, I want you to always feel comfortable in chatting with someone on our team to be able to do so. Again, in terms of your evaluations on this side, we have a midterm for you. Again, we want you to do well in that. It's not to try to get students out of the course. It's not to have a certain average. In fact, we want you to learn from this experience in a lot of different ways. We're going to ask you to post your sample questions and sample answers online so that you can actually practice your questions before you actually write the term test. And we want you to have the benefit of being able to post, see other people's answers before you actually write the midterm or even the final exam. Hopefully you're not going to lose your sanity along the way. Again, that's my job to make sure that everything is going well for you. So the term test is basically going to be multiple choice and short answer for both the midterm and the final. Final exam won't be cumulative. We'll be covering a lot of primary papers, but you don't need to read those papers. I want to present those papers to you and for you to take the most important things out of those primary papers for you to learn about on your own time. Any questions about the test and final exam? How many of you enjoy taking term tests? It's like your favorite thing. Honestly? Yeah? Yeah, okay. Would you be willing to take 120 of them? Take one for everyone in the class. No. Okay. So I know for some individuals the term test can be like really, really stressful, and it's stressful for me too watching you write the term test, to be quite honest. And I don't want it to be a really stressful event, so we'll be trying to prepare you the best that we can, but this is the format just to prepare you in advance that it will be multiple choice. We'll be giving you practice tests, and we'll be giving you questions so that you can practice them ahead of time. So even though it might be stressful, we're going to try to lower that stress for you. In terms of studying, one of the things that I'd like you to maybe practice is something that you learned in your PSL 300 class. You guys have all taken PSL 300, right? Yes? What do you remember the most from PSL 300? Just out of curiosity. Dr. Key, okay, awesome. I'm not Dr. Key, by the way, in case you're wondering. Anything else other than Dr. Key or Dr. Tweed? Yes. Okay, Dr. Tweed was really interesting. Awesome. Do you guys remember by any chance this step cycle of locomotion? Yes? Did you love that step cycle of locomotion where you had these little central pattern generators and the... No, didn't like that? But actually it's a really important concept for you because one of the things that happens is when you're walking, you actually don't think about walking, right? So you do it. It's like a central pattern generator that you've learned about. And this actually happens to use what's known as a feed-forward mechanism. So you guys have written lots of multiple test choice questions. You guys have maybe written short answer questions. And so you're using this prior experience just like walking. So you're used to walking, you don't even think about it. So you're going to be studying for these things in the way that you've done in the past. But now you've gotten this brand new course and you hear about all these different things that are going to happen to you in this course. And so you're going to have to adapt just a little bit and you're going to have to use these feedback mechanisms. So if, for example, you started walking and there was a little divot in the ground, you'd have to adjust your step cycle of walking to make that little divot in the ground or that little um, uh, slope in the ground something that you take into account. And you're going to have to adapt a little bit in this course. And my job is to help you to adapt in this. So you've heard a lot about active learning, and I'll get back to this in a moment. And this is part of that feedback mechanism, learning to adapt and learning how to share your answers and learning how to adjust your answers based on what you see your colleagues writing about as well. And you'll understand this when that time comes around. And what I'd also like you to do, because it helps me a lot, it's really hard to remember everything from a lecture. So when you leave here, and I'll show you this in a few moments, you're not going to remember the vast majority of what you learned in today's lecture. And that's just a natural consequence of how our memories don't actually solidify in our brain really easily. But I'd like you, wherever possible, to use what we call the 3x3 method. So I want you to think like me. Right? That's ultimately one of my goals anyways, that you're going to be thinking of like 3 different slides that you might think are going to happen or show up on the lecture. I also want you to think about 2 different things. So that's the first 3. And then the 2 other things are 2 items that were confusing for you, and then 1 thing that you never knew before. I can't really ask or I can't really answer questions when students come up to me on the street or come to my office and say, I really didn't understand slide 17 from lecture 3 that you covered on January the 27th. My brain doesn't work that way, but my brain does work in a way that, remember that lecture that you were giving where you talked about the gut microbiome and how it causes Parkinson's disease, or how the gut microbiome is changed in people with Parkinson's disease? I really didn't understand all of those different bacteria that you were talking about and why that's important. That's the way that I'd like you to maybe think about talking to me about some of the issues that you're having with lecture. The other thing I want you to do, and I hear this all the time, so I want you to maybe change your focus just a little bit. You're here in lecture, or you might be listening to this lecture from home, and that's great. And I think that that's a good way to start learning this process. But if you're in class, and you're just in class, and you're sitting and you're listening, you're not going to take away very much from this. And a lot of times students have this idea or this notion that that's all you have to do, that you have to come to class and review your notes and then you'll get perfect on your final exam or on your term test. That's absolutely not true. And what you need to be able to do is to find ways to engage the material, learn it in different ways, and to share your knowledge in different ways with other individuals in class. Because I want you to again, connect in this course. I want you to interact with other people in this course, and I want you to really learn things in this course. So let me give you an analogy that I like using, and part of the reason why we're going to talk about food addiction is it turns out that I'm addicted to sugar. I didn't know that I was addicted to sugar, but I have all these cravings right now of like running out and buying a chocolate bar and sitting down and eating it right in front of all of you. And that's the sort of thought that's running through the back of my mind as I'm up here talking to all of you. But imagine this. So imagine you have you've gone out and you've bought this extra large pizza and you've like loaded it with all the favorite toppings that you've had, right? And then you have like 1 hour to eat the whole thing. Would you be able to do that? Or let's say I make it even worse. I give you 50 minutes to eat the whole thing. So it's probably not a good thing to do and cram all of that in one sitting. And it's probably better to space out how you're consuming or how you're digesting whatever food item that you've purchased. And I want you to think about the course in the same way, that if you're leaving things, and you've done this in the past where you've crammed, this isn't a course that you're going to be able to get that A or that A+. If you're spending time every single week, then this is the course where you can get that A or that A+. And that choice is up to you. I'm not going to be able to come to each of your homes and to make sure that you're actually doing well. This is what I don't want to happen to all of you. I don't want you to have a Mariah Carey moment. I love Mariah Carey, by the way, so I'm not making fun of Mariah Carey. I happen to have all her CDs. And you know she's a professional, and you know that she's a very talented individual, and you know that she's got this long history of success in the music industry, and all it takes is like one instance to mess things up and to completely ruin a great singing career. And I don't want that to ever happen to you. I want you to be well prepared and to be thinking about this course in a way that you can prepare for it, get that A or A plus in this course, but that's all up to you. Whether you become a Mariah Carey or you become like a superstar singer in a different way, that's completely your choice. So on Friday evening, I'm going to be giving you a weekly study outline. It won't be posted on the announcements page. It'll be a separate box for you. All you have to do every single week is to find time to go through and check those things off. That's all I want you to learn from that week's lecture. You'll get one this Friday, for example, telling you what you should know from this lecture today. All of the lectures are going to be provided for you. They're going to be either on YouTube or on social media. You can get involved in these recognized study groups. It's a great way to get involved and to engage in active learning. And your success is really, again, important for me in this course. This is Ebbinghaus, who is a psychologist who showed very clearly that as soon as you walk out of this classroom, if you don't think about it and you start thinking about other things, you're going to start forgetting the vast majority of what you learn within the first 30 minutes to an hour. And the only way to prevent this type of decay is to actually review on a consistent basis. So Friday night, on the weekend, go back and review your notes so that you don't feel like you're having to cram or eat that entire pizza at the same time. So when we're going through some of the different topics, we're going to be looking at things like TMS. And how many of you have heard of TMS? Just again out of curiosity. You don't count, but that's okay. Sorry, you do count, Kathleen, but I know that you actually know a lot about TMS. And how many of you have heard of DBS, deep brain stimulation? Okay. And I know that many of you have taken courses related to the medical imaging of the brain, and we're going to be covering those different topics. We're going to be learning a lot about the prefrontal cortex this year, and then we're going to be looking at some of the limitations as well as advances in molecular cell biology in 2017. So I'm going to come back to this question in a moment, and then we'll take a break as you think about that question, about how would you improve the brain. Again, you've had a chance, hopefully, to look over the course syllabus, and this is sort of the breakdown for the course syllabus. There's one modification that I want to make sure is okay for all of you, and I'm going to bring that up right now. And if there are people watching online and you have some questions, now is the time to jump right in. Kathleen, do you not have access to the Blackboard site? Okay. But by the way, these are the two TAs that are in our course. So this is Kathleen and Darren. So they will be helping to give some of the lectures. They'll also be providing you with feedback on assignments. They're the ones that are going to be helping with some of the evaluations. They're great resources. They're great individuals that know a lot about the systems that we're going to be talking about, and I hope that you'll find the time to chat with them as well. So again, I'm going to offer this course up as a very flexible marking scheme. So outside of one of your colleagues, is it Armin? It's not Armin. Sorry, what's your name? Yes, you. Oh, Adnan. Outside of your colleague, who loves taking, sorry, I can't hear well with the headphones on. So outside of your colleague who loves taking term tests, some of you may not really like taking term tests, and you might really enjoy putting together like a multimedia project where you do a little bit of writing, find links on social media, or find other videos, other sort of infographics, etc., and then put them all together as a multimedia assignment. Some of you might feel like you're going to be doing better on that than you are on your term tests. So we want to make it flexible. So if you get a really great mark on your term test, that'll be worth a little bit more. If you do really great on your multimedia assignment that we'll be going through in some more detail later, then that will be worth more. So in either case, we're going to make sure that you get the better mark, and the mark will be weighted in your favor depending on which assignment you do better on. You're going to find one primary paper, and you're going to be creating an infographic that I'll talk to you a little bit about, as well as that final multimedia assignment. It'll all be related to this one paper of your choice. I'm not going to pick one for you. I can help you find a paper. You can ask Kathleen or Darren to help you find a paper, but I don't really like boxing students into a topic that I want you to cover for me. I want to see what interests you in neurobiology. We're going to have a number of different online participation assignments. They'll be fairly easy to do. Again, the hallmark there isn't for you to get low marks, it's to really help you study. I'm hoping that in this class this year, the average mark on the online participation, as it was in the fall semester, was 12 point something out of 15. I'm hoping that it's at least 12.5 or higher on the participation, so I don't really think you'll be too stressed out by this. The last thing I'm going to mention is this more flexibility. 35%. Are you guys comfortable with a 35% final exam? Perfect? Yes, love 35%. Compared to other courses, 35% may not sound like a lot, right? Okay. But one of the things that I want to do is I'm going to create a series of online quizzes. And these quizzes will be helping you study and to prepare so that you're not cramming and that you're at least reviewing the material on those quizzes week by week in the last sort of 5 weeks of class, getting ready for the final exam. And you can decide that you want to write 5 of those, and you'll get a mark on that. And again, it's not designed to lower your mark in any way, and this will be added back to your participation marks. And at the same time, one of the things that will happen is that it will also reduce the weight of your final exam. So I've done 5 online quizzes, and I got perfect on all of those. So I'm just going to take 5% off of your final exam. Does that make sense to you? So you're going to have 5% reduced from the weight of your final exam. But wait, what if I only do 1? So we'll just take 1% off of your final exam, and if you do 2, we'll take 2% off, 3%, etc. Do you have any questions about that? You don't have to do any of them, by the way. The exam will be exactly the same for everyone. If you like living dangerously and you just want to do a 35% final exam and you want to go for it, please do. Yes? So it's automatically reworked? So like at the end? Yeah, all of this is done automatically. You don't have to tell me that, yeah, you know, I'd like to have a little bit less on my final exam. Or you don't have to tell me that you want your midterm weighted more, etc. Yes?
Just for clarification, let's say I do a quiz and I do horrible on it. Do I still get the percent, or do I get like 0.5% or that 0.2% or whatever?
So I think one of the things that's going to happen, and this was a suggestion that one of your colleagues made, and by the way, we're still looking for people to join the course management team. We would like more people. The course management team suggested this, that you can take the quiz that might have an open window of about a week. You can do it multiple times until you get the mark that you want. Does that make more sense for you? Any other questions, suggestions, comments? Yes.
Is there like a list of deadlines for the participation assignments?
Generally speaking, they're clustered the week before, like the term test, or a couple of weeks before the final exam. But again, it's not going to be onerous where you're going to feel like there's a lot being asked of you. So they're fairly short. They'll take about 10-15 minutes. Yes?
In the syllabus,
Yes.
it says February 16th for the term test and February 9th. But on the actual, when it has the dates, it says that it's on the 9th.
I'm going to go back and check the date. It's either the 9th or the 16th. I have a couple of different classes that are running around the same time, and I might have gotten some of the dates mixed up, but I'll confirm that date with you. Okay? Any other questions or things that you need clarified? No? Yes, way at the back.
What's the multimedia component?
So I'm going to be asking you to do a little bit of writing. So you're going to do things related to future directions. You're going to write out some experiments or write out some suggestions for what these authors might do. And then I'm going to ask you, instead of doing a literature review, I'm going to ask you to find sources online to help present some of the background information related to the paper that you're covering. So if it's on Parkinson's disease, rather than just writing up all the signs and clinical features of Parkinson's, you might actually find videos, or you might find other infographics or other sorts of summaries that you might be using on this one sort of page that you'll be creating. Okay? And again, I'll make that a little bit more clear for everyone in class as we get a little bit closer to actually deciding on papers, etc. So again, I'll go through this in more detail, but some of the things that we've been asked to do with our students is to change the way in which we're getting students to do assignments. And some of this is going away from traditional write out like a 4-page, 5-page literature review. You're still asked to do a lot of that in grad school, but actually even in grad school you're asked to create more and more visual abstracts and visual ideas. And so we're going to be asking you to be more visual in your presentation as well. I will be giving you workshops and going through the expectations of this prior to the actual start of the project, the rubric. And as I tried to do with the fall semester, I'm actually going to do the assignment along with you, so you get to see what my assignment looks like. And of course, both Kathleen and Darren will give me 100% on the assignment, so you can use mine sort of as a rough guide to what the expectations are going to be. You weren't intending to give me anything lower, were you? No. Okay. Awesome. So you'll see like what sorts of things I'm doing, the types of things that you might want to do on each of the assignments as we're going through them. So again, I'm not going to go through this in a lot of detail because we're getting pretty close to having to go to break. But one of the things I want you to really do, if you're just coming to lecture or you're just reading through your notes and this is all you're doing every week for review, you're not going to do well in this course. I can guarantee you that. I can guarantee you that if you're going down here and you're talking to your colleagues and reviewing as a group, or you're spending time even on social media and comparing answers, etc., you're going to be doing a lot better in this course. And you need to get away from that model of sitting at home and just reading through your notes and reviewing your notes only. That's not the type of course this is. It'd be more akin to what you would do well in anatomy or other courses where it's just pure memorization. So some of this also gets back to, I want you to really do well outside of this course. I want you to get into those research labs. I want you to get excited by doing research. I want you to go think about med school or dentistry or professional healthcare, or I want you to think about what you might do afterwards. And the only way I can write you a really strong reference letter, which is outlined for you here, this is typically what's asked on a reference letter, is to be able to see that you've been engaged in things where you're working with other people in class, and you'll understand that some of those online assignments actually require you to provide some feedback to someone else's answer online in a very collegial manner. The way that you communicate, whether visually or on a test, etc., all of this is related to be able to write you an excellent reference letter also at the end of the course and say, wow, you know, this student did all of these things incredibly well. I'd be able to rank them without any difficulty in the top 5% of their entire cohort. And that's really one of the things that I love to be able to do at the end of this course, is to say that you're an excellent student, that you should be going on to do other things after the end of this course. Because we're consistently reinventing neuroscience, and actually this becomes part of the segue that goes into the second part of the lecture, which you are responsible for. Are any of you taking a microbiology course? Can you tell me what's shown to you here on the left? And be specific as possible. Is it like a staphylococcus? Bacteria? So do you agree with your colleague that it is bacteria? Anyone disagree? Everyone's in perfect agreement with you. You mentioned staphylococcus. Why staph?
I'm just trying to remember which one looks like a string.
Which one looks like a string? Okay, so great. So it's either strep or staph, and you're right, it's a coccus. So it's a small round-shaped bacteria. So each of those little balls that you see there is a bacterium, and you're absolutely right. So why are these bacteria related to you washing your hands? Keeping your hands clean, always a good idea. Any other ideas in terms of how this might be related to behavioral biology? do you know of any condition, whether it is a neurodevelopmental condition or whether it is an affective condition, that there's a lot of hand washing involved? OCD, right? And that's one of the things that we stereotype as obsessive compulsive disorder. And it turns out that this theory that was developed in the 1980s, which is called PANDAS, which is why you see this here, is that if you are exposed to streptococcus, so good guess, by the way, if you're exposed to streptococcus when you were young, that you would develop things like OCD because of the effect of the toxins that the bacteria have on your nervous system. And for a while, for about 20 years, this idea and this theory was actually tossed out the window. There can be no possible relationship between early bacterial exposure and the development of a neurological condition like obsessive compulsive disorder. But now we've actually come full circle, and we now accept that bacteria can very heavily influence different types of behavior as well as neuropathologies. And that's something that I'd like you to maybe be thinking about. And the last thing before we take a break, and we'll take about a 15-minute break, so if there's any coffee left at the back, please help yourself. But here's a little challenge that I will take up the answer with you after the break. If you wanted to make your brain the best that it possibly could, to be the best student brain that you possibly could make, what are the different types of things that you would want to try to get that person or individual to do to make this super brain? And that's something that I'll ask you, the answers that you got after the break, and we'll take about a 15-minute break now. And please help yourself to coffee, tea, or take a quick bio break. there is a salmon-colored sheet going around. It's just if you want to fill it in, don't put your name on it. It's anonymous. It's just to see how I can help you in this course, etc. No marks assigned to it, nothing. It's just, again, to let me know if you're feeling stressed or if there's anything that I should come back and address in the next class. Thanks.
Professor, do you have an extra mic too?
There are some at the back. Yes, I'll get one for you.
No, I'm sorry. I saw the rooms there.
You'll see the same thing on your
It's right past the stand.
Oh, the packet one. And wait, oh, no, no, no. Oh, does it really matter? That's one that that's the one I know. The only one I know. Okay. What is the and the guy who got
I think so. Is it like that?
I was 12 in grade 7 when we watched this. So that's how many years ago. Because I remember I was like I was 13. Like you were like no, that would be crazy. At in front of Starbucks, but that's at the front it was like start. What else good Lava House is I felt like such a loser. It's my deviated from
Okay, folks, that white sheet with the crossword is actually more for you after class if you want to go back and review it. Some of those terms are ones that you're going to need to know for your upcoming term tests, so it's just sort of a different way to reinforce those ideas. We want to have a little bit of fun in this course wherever we can, and so if you can do that in your spare time, that would be great. And I'll leave the folder if you want to leave your salmon-colored sheets with your short answers. If you have anything that you've written down, please don't put your name on it or anything, and you can just put them in anonymously at the folder at the front. So I'd left you with this challenge. You want to create the best possible learning brain, like a super brain that can learn at the drop of a hat. What would you do? What kind of brain would you develop? How would you develop that brain? Any ideas? This is going to be a long semester. Yes.
I don't know. Sorry, I don't know how, but I want my brain to be able to take down any like self-enforced or like environmentally enforced barriers that prevent me from like full like productivity. Like
Okay, so being able to concentrate
yeah.
better. Okay, awesome. So a brain that can really stay on task and concentrate well. Great. We don't know how to get there, but that would be like one feature of a brain that we might want to have. What else would you want to have as a super brain devoted and dedicated to learning? Yes.
The brain that can run on less sleep.
Sorry, that can run on
Less sleep.
less sleep. Okay, actually, I think that's going to be really hard. People have been trying actually to figure out a way to let you get by on less sleep, but in fact, it's been one of my ideas to try to get students to actually sleep in class. I see a few people have already taken me up on that offer, but it's actually not a bad thing. Sleep is good for your brain, you're right. Could we get away with trying to develop a brain that requires less sleep? It's a really intriguing hypothesis. Anything else that you guys have thought of? Yes.
I'm not sure if this is still a myth, but is it that we use only 13% of our brain at once, or is that still
That we only
access more of it at once?
to access more of our brain. So I think I was just watching that movie over the holidays,
Yeah.
and I forget what that movie was with the lady who's on the Avengers.
Oh, Scarlett
Scarlett Johansson, Lucy, yes. And it is a myth. We actually use a substantially larger portion of our brain than 10%. And it's one of those things that I'm hoping that as you read more papers, that those types of myths are ones that as neurobiology students that you guys also start to overcome as well. So let me tell you a little bit about why I'm interested in Gwyneth Paltrow, and we won't tell my wife why. But one of the things that she did in 2010, she actually had a movie where she was a country singer, which is why she has a guitar there. And one of the things that she had to do for her role was to actually gain 15 pounds. I don't know if she looks overweight there, but she gained 15 pounds for her role. And then after her role was finished, she had a lot of problems. She couldn't take the weight off easily. She started to suffer from a number of emotional as well as mental health breakdowns. And she felt that it was the weight gain that actually precipitated both her physical as well as mental health breakdowns. Of course, I like eating. You can tell I like eating. I'm designed to eat. I eat all the time. I like eating practically anything that you can think of. And it's gotten to the point now, specifically last year, in about March or April of last year, I had my first stroke. And my first stroke was something that I didn't even know had occurred. So I started losing vision in my right eye. I couldn't coordinate the right side of my body. And I thought because it was a time where I had the flu that it was actually related to the flu. And it was a micro stroke. I was really, really lucky. Again, I'm not obese, but I am overweight. And this, again, it's not where I'm exercising regularly. I'm not incredibly unhealthy, but nor am I healthy. And again, I've taught a lot about stroke, and we're going to talk about stroke in this course. And I didn't even realize that those are the same symptoms and signs that I've been teaching that I had in my own sort of brain. And because I know that I have to change the way that I approach my lifestyle, otherwise my next stroke is going to be incredibly bad, that I have to change a lot of things about my lifestyle. One of the things that's been really hard has been to change the way in which I eat, because I like food, as I mentioned, and food is something that I thought would be really easy to change my habits of eating. And it turns out that our eating habits are somewhat ingrained in the brain, as well as changing some of the gut microbiome. Both of those things, the overeating because of a behavioral change in a circuit in the brain, as well as its long-term consequences and its impact on the gut microbiome, which contributes even more to the unhealthy brain, are important factors that now is emerging as a topic in neurobiology. I don't need you to go back and to look at these papers, although I think that they're really interesting papers. And I'll mention them in passing. You don't have to learn anything about them. I'll tell you what you need to focus a little bit on. But if you look at weight gain, all of you probably ate a little bit more than you normally do during the holidays. And when you gain this weight, you're not just gaining weight, you're actually changing the gut microbiome, the resident bacteria in your gut, as well as setting yourself up to make it harder for you to actually lose that weight that you have gained. So some of you will know that once you've gained weight, just like I have, that it becomes harder and harder to actually lose that weight. And we're going to talk a little bit about this later on. And these are fairly new papers that came out in late 2016 and early 2017. And we'll be going through some of the aspects of these later on. And what it basically says is, once you've established an unhealthy diet, it is really, really hard because of the changes that occur at the level of the gut microbiome to go back and lose that weight. The gut microbiome itself changes, your behavioral circuits change in the brain. Both of those are working against you to make it really challenging for you to lose that weight. And that's the challenge that I have right now. So, I'm not too advanced in years, although I'm older than you by quite a lot. Bless you. And one of the things that I want to do is to be able to have the opportunity to play with my grandchildren one day, and I'd like to be able to make it to that age. And I know that if I don't change my behavior and I don't change the way that I live, that the next stroke is going to dramatically affect what my lifestyle might look like. So I'm going to go through, maybe for some of you, some new ideas and some concepts. I'm going to talk to you a little bit about gut adaptation and why we talk about the gut and why we think it has such a big impact on the brain. I want to also understand how our behavior, when we eat things, how we are actually not just gaining weight, but we are also physically changing the resident microbiome, the gut bacteria that lives within us, and that's increasing the likelihood that you're going to be changing your weight as well. Eventually, and I'm going to try to make this case for you. So today I'm just giving you the background. I know a lot of you aren't, who are listening at home, you're still on your way back to Toronto, you're still on vacation, and a lot of you are still just slowly getting into the mode of things. So today I'm just giving you the background on the neural circuits that you probably learned in your second year, as well as an overview of the gut microbiome, but eventually we're going to be looking at parts of this paper. And when we look at Parkinson's disease, is there a correlation between your gut, so something that occurs in the bacteria in your gut, and Parkinson's disease? And it's again, a really interesting hypothesis if it turns out to be true. And again, it was published fairly late last year in December of 2016. And it's a paper I'd like us as a class to think a little bit about. Can we actually impact a neurodegenerative condition by our diet? It's a radical idea. It's a radical hypothesis. Your ability to change your brain is not just resident on neuroplasticity. It also is important in terms of the gut microbiome and how it regulates your brain health as well. So for some of you, this will be background, and for some of you this will be new. If you go through that crossword, the key terms that I want you to make sure that you know, things like dysbiosis, the gut microbiome, are actually emphasized there for you. I'm not big on numbers, by the way, so I don't need you to memorize numbers like 10 to the 14th or 10 to the 10th. It's just a trend or an idea that I want to get across for you. We have living, residing on us as well as inside of us. So your skin has a microbiome, and it's called the skin microbiome, and you have a number of bacteria that are living on you all the time, and most of the time it makes no difference that you have them. You also have bacteria that has colonized your gut by the time that you were born, and that gut colonization is what we call the gut microbiome. And we have a lot of these bacteria internally. Again, the number, not something you should memorize, but 10 to the 14th bacteria inside of our guts. It's now thought that this gut and the microbiome in general, or all the microbiota, all of the living organisms, fungi, bacteria, viruses, etc., contribute to our health status as well as our disease status. You're healthy not just because of what you're eating, not just because you're sleeping well, not because you're just exercising, but also because of your gut, skin, microbiome, etc. All of those are actually interrelated, which is one of the themes that I'll be talking to you about. All of the genes that are encoded by the various bacteria, viruses, etc., is known as the microbiome. The microbiota are the actual organisms, and all of the genes that are encoded by those organisms is known as the microbiome. Can you think of a reason why people are interested in the genetic expression of the bacteria that live in your gut as well as in or on your skin? Any ideas? Why is that so important? There are two concepts here. Any ideas? Any guesses are perfectly fine. This is, your classroom. This is your opportunity to talk. Yes?
To see if there's a difference between how they are expressed in the gut versus the skin.
Sure. So we know that there are different bacteria that live in your gut versus your skin, absolutely, and they express different genes, absolutely. So what do you think is easier to do? Is it easier to change your genome that you're born with, or is it easier to change the genome of these different bacteria? Different bacteria. It's a lot easier to change the bacterial genome than it is to change your genome. Which of these numbers, and again, I don't put the numbers up there, but which number of cells, your cells or bacterial cells, is bigger? Do you have more cells than you have bacteria living on you or in you? So we think you have about 1 to 3 trillion cells throughout your entire body. It's just a guesstimate, and we have a lot more of these bacteria. So not only do we have a bigger population with a bigger genome, with more genes, but we can modify those much more quickly than we can your genome. You're born with your genome. We can turn it on and off through epigenetics that we'll talk about much later in the course. But if I want to make changes to your health status, it's through these bacteria that I can modify relatively quickly. And again, it's not something that is a totally new concept. If we change either through diet, the types of food that we eat, or through the types of healthy lifestyle, we know, for example, in 2017 now, that individuals who are much healthier and live a much more aerobic or much more exercise-driven life have a very different internal gut microbiome than those that are sedentary or those that don't exercise. So we know lifestyle, like exercise, we know that diet, whether or not you have clean hygienic practices or behaviors, whether or not you were exposed to antibiotics at an early age. All of these things will impact your intestinal microbiome. This can lead to things like metabolic dysfunction. So you've heard of type 2 diabetes, where you have like pre-diabetic status. We now know that changing the internal microbiome can lead to things like metabolic dysfunction. We know a lot of the things that are bad for the brain actually occur because when your bacteria in your gut change, so do the chemicals that get released as a downstream product. And one of the things that happens is that you have more inflammation throughout your body, particularly in areas like your brain. Your brain is very, very susceptible. If you haven't learned it in your second year, your brain is very, very susceptible to cytokines that are inflammatory markers. By changing your diet, you change your microbiome, you change the level of cytokines, which can impact on your brain. And we'll be coming back to different types of cytokines later on in the course. And you don't have to memorize these right now. It's just to give you an idea of why people are interested in this microbiome. This concept that we can have an alteration in your normal gut bacteria, the things that keep you nice and healthy. If we change that and make it a different population of bacteria, we can change your health status. This is this idea of dysbiosis. So again, most of the gut bacteria, which makes sense, you don't have air down in your gut, you don't have it in your small intestine, you don't have air down in your large intestine or your colon. Most of the bacteria that you have now, this was not the case early on, but all of the bacteria that you have now in your gut are anaerobic. And these anaerobic bacteria that live there, and we're just talking about the bacteria, we have about 500 to 1,000 different species. So we have a lot of variation in the types of bacteria that can live within your gut. The most abundant phyla, so the larger category, so even though we have a large number, 500 to 1,000 different species or distinct bacterial species, most of these are either gram-negative or gram-positive types of bacteria, which are either called the bacteroidetes or the firmicutes. And it's the balance between these two, whether we have more gram-negative or more gram-positive, that cause changes in your health status. And that's one of the things that I'd like you to make sure that you know. When we talk about the gut microbiome, and we talk about the gut in general, because of the way that we eat food and the way that you have had your intestinal tract colonized by this bacteria, we see remnants of this colonization process. So for example, and again, you don't have to worry about all of the details here, especially the numbers here on the right. I'm not great at memorizing numbers myself, and therefore I feel that it's really inappropriate to ask students to tell me back the numbers that I myself have to look up here to actually see. The numbers here not being important, but you can see the overall trend as we go from the stomach all the way down to the colon, that the number of different bacteria and the overall number of species of bacteria that we see here on the left actually increase. We have very few bacteria, relatively speaking, in the stomach, by the time we get to parts of the small intestine like the duodenum, the jejunum, and the ileum, we have greater numbers of these bacteria, and we also have different types of bacteria, the lactobacilli, as well as the streptococcal types of bacteria living within the small intestine. And by the time we get to the gut, the one that I'm talking about, this is when I'm talking about the gut microbiome, I'm talking about the colon. The colon has the largest population, the greatest number as well as the greatest diversity of all of the bacteria that we have. So this is because of the way in which our gut was colonized. So if you go through and take a look, early on, we have no bacteria. As far as we know, no bacteria lives within your gut normally. As soon as you are exposed to the environment, the bacteria start to colonize these different areas, and some of these bacteria are more aerobic than they are anaerobic, and we have more anaerobic bacteria the farther down that we go. Do you have any questions on this before we go on? Are you guys good memorizers or not so good memorizers? Or outstanding memorizers? Outstanding? Sure? Avinash, I think it's going to be an awesome exam for you. Um, so I'm not a great memorizer myself, so I'm going to ask you to memorize a couple of things on this slide, which is the diversity from the stomach all the way down to the colon, an increase in number as well as an increase in diversity. And I am going to ask you to make sure that you know a couple of the bacteria, because we're going to see these multiple times. That we see a lot of lactobacilli up here in the stomach, and the lactobacilli stay in the entire digestive tract. So we have lactobacilli in the stomach, in the jejunum, duodenum, and ileum, and we also have lactobacilli down here in the colon. So we have it in all areas of the gut. As well as more diversity that we're going to talk about here, but you don't need to memorize all of these other bacteria for the time being, okay? So on this one diagram, it's really just about the increase in numbers, the increase in the diversity, and the fact that some bacteria like lactobacilli are consistent all the way through the digestive tract, okay? So again, as I mentioned, we have these two different phyla. Even though we have about 500 to 1,000 different species of bacteria, if we were to separate them out into the larger classification, the phyla, we would actually find that most of them fit into either the bacteroidetes or the firmicutes. The other interesting fact is, even though we have a lot of this diversity around and we have a lot of this differentiation around, each of you has your own unique fingerprint of a microbiome. You have your own unique species of these bacteria, and this is what is known as an enterotype. The types of bacteria that dominate in your gut actually are somewhat unique to you. Now, I say somewhat for a variety of reasons. Which do you think plays a bigger role in this differentiation or diversification? Do you think it's genetics? So if my brother who is living in another country, for example, and we are genetically related, are we going to have a closer enterotype, the type of bacteria that live within our guts, or is my wife, who is not related to me directly through genetics, but we live in the same geographical area, are we going to have the more similar enterotypes? What do you think? My wife? So the environment tends to be the more important driving factor here. That the types of food that we eat, the types of stressors that we're exposed to, it's not necessarily the genetics that drive the gut microbiome. And we use that to our advantage, because if it's only genetics driven, we can't really change that. But we can change the gut microbiome by changing diet as well as changing some of the lifestyle factors. So again, the dominant bacterial enterotypes that exist in most individuals is a balance between the bacteroides, Prevotella, as well as Ruminococcus. So all of you have these three dominant types of bacterial enterotypes that are unique to you, But some of you will have more of the ruminococcus, some of you will have more of the bacteroids, and some of you will have more of the prevotella than your neighbor sitting beside you. But it is a way for us to go back and fingerprint your individual enterotype or your gut microbiome. Your lifestyle, as I said, and this is sort of the first module that we'll be covering. So this new idea that the gut microbiome that we have can be influenced by things like our diet, things like exercise, things like your sleep. So I think one of your colleagues had mentioned earlier, could we actually get by on less sleep? The reality is we can't right now anyways. And so all of these things not only affect your brain, but it also affects your gut microbiome. So again, you don't have to memorize this diagram. We're going to come back to this diagram several times over this module, and I'll talk to you about this in some more detail. But if you are healthy and you're a healthy individual and you are balancing things in your lifestyle like your diet as well as your exercise, the amount of sleep that you get, your microbiome stays relatively healthy. It doesn't change very much. It changes when you go from being an infant to being an adult, and then when you get into older age, as you start to get to 60 years of age and older, your gut microbiome starts to change again. And again, think a little bit about this. If your gut microbiome is changing by the time you're reaching 60 to 65 years of age, when does neurodegeneration take place? For most cases, and again, it's correlational, it's not causational, it's just a really good sort of symmetry, that we tend to see neurodegeneration where we don't have a genetic cause in ages where they're somewhere between 50 to about 65 years of age. So again, this is one of the reasons why neurobiologists are fascinated by this concept of the microbiome. So the stability of the microbiome, when you're nice and healthy, keeping a nice and healthy, stable microbiome is really important. We know that changes in your lifestyle change this balance that we're going to be coming back to, the balance between the bacteroidetes and the firmicutes. If it changes, so does your metabolic status, and therefore so does your health status. If you take different drugs, antibiotics wipe out all of your microbiome. It takes an awfully long time. Let me ask you, and you don't need to write this down, but I'm just curious how much microbiome backgrounds all of you have. If I took an antibiotic and I wiped out all of the bacteria that live in my gut, how long does it take for the pattern that I had before I took the antibiotic to come back? So let's say I had a really bad chest infection. I started taking antibiotics for it. My chest infection cleared up. Then I go back to what I was doing before. How long does it take for my microbiome to reestablish itself? Yes? It never does. How about as close as possible to what it was before? How long do you think it takes? A week? A month? A few hours? Okay, that's an interesting hypothesis. So it takes somewhere between 6 months to a year, depending on your age, for your gut microbiome to go back to where it was before you started taking antibiotics. And we're not really sure what the long-term results of all of that are. So we'll talk a little bit more about the stability of the microbiome later. And again, you don't have to memorize all of the features that are on here. It's just to give you some background information on what we're going to be talking about. So this idea of dysbiosis, which is a definition that I'd like you to be very comfortable with, familiar with, and to be able to give back to me as a definition, is the imbalance in that normal microbiome community within your body. Either through antibiotics or a change in lifestyle, you enter into dysbiosis, you enter into this imbalance. And it's a concept in microbiology. So a time series showed that, again, for most healthy individuals, all of the bacteria, the viruses, etc., stay very, very stable while you are healthy. However, changes in your dietary status, changes in your environment, other types of things can dramatically affect the normal gut microbiome and cause you to fall into this period of dysbiosis. Again, it's not just the bacteria, and we talk a lot about the bacteria. It's been much, much more challenging to actually tackle the gut virome, all the viruses that live in your gut. We're just starting that now. Even though we've known about it for 10 years, we're just now starting to look at the gut virome in the way that we do. Can you think of a reason why, going back to what we talked about, we're going to cover many different aspects of neurobiology using a number of different techniques. Can you think of a reason why it's been incredibly difficult to actually find out what viruses live within your gut? It actually was really challenging just to figure out the bacteria that live in your gut. Any ideas? So we're going to come back to this idea a little bit later on, but in order for you to actually grow viruses, you need a host vector. They don't grow by themselves. They have to infect tissue, etc. And it's been really hard to find a suitable vector or cell for them to infect for us to be able to go back and sequence all of the different gut viruses. But again, as far as we know, they tend to be just as stable. So how does things like diet, because that's my primary focus over this next little module, how does diet actually affect the gut microbiome? So this has actually been an area of study. When the first ideas that maybe your gut and the changes in your gut actually change your health status and your mental cognitive abilities, possibly things like affective mental health disorders, etc. That was back in the 1990s. No one actually believed those initial papers that came out, and it wasn't until the mid-2000s that individuals like Turnbaugh and others like Wu came up with the data because of technological advances in sequencing that allowed us to go back and look at the gut microbiome. So if we look at a low-fat or a healthy diet that is rich in plant polysaccharides, so a nice healthy diet, right? So those of you who chose the Oreo cookies at the back, poor choice. Those of you who chose sort of the vegetarian or the vegan option of the breakfast bars, better choice, because we're looking at low-fat, plant polysaccharide, fiber-rich diets versus having a high-fat and high sugar content, or the westernized diet. So again, think of my diet and you'll have a really good idea of what that looks like. Heading off to McDonald's and then getting that quarter pounder and that small drink that you get, which is about this big, and the fries and all of that high fat, high sugar, westernized diet, they were able to change in mice the gut microbiome in a single day. Now, we're not talking about humans, although there's a paper that follows this that does look at humans, but in mice, changing the diet was able to change the gut enterotype, the fingerprint of the types of bacteria in one day. Think a little bit about that. One day is enough to change this. And if the gut microbiome is all that important, then this becomes a really big factor. This study, by the way, by Wu et al. In 2011, when they examined the gut microbiome in individuals who were exposed to a healthy diet versus ones that were then exposed to an unhealthy, high-fat and high sugar-containing diet, they were able to see similar changes in the human gut microbiome. But here's the one caveat. This one caveat is that this study has not been replicated, and it's been about 6 years or 5 to 6 years now. And we have a lot of studies in mice, but the studies in mice don't necessarily translate to what we see in humans. And again, some of this is really challenging and technically challenging, and I'll get into that when I go through the paper in the next lecture. So this study by Wu et al., which confirms the study that was done 2 years prior by Turnbaugh, shows that there is a correlation between diet and in a high-fat diet, these bacterioids tend to dominate. In a carbohydrate-rich diet, different types of bacteria like Prevotella dominate. So in my system, my enterotype, because I have a high-fat diet, I have a high-sugar diet, I love sugar. If you could hook me up to an IV machine with nothing but sugar, I'd be the first one in line. The most embarrassing moment I ever had, over the past couple of weeks, which actually led me to try to reduce my sugar intake, was my wife knocked on the door to my office down in the basement, and she caught me like red-handed with like a Twix bar in one hand, and I also had like a bag of chocolates in the other hand, and she caught me like mid sort of eating, and she was like, "What are you doing? Like, do you not realize that you're like destroying your body?" And I said, I'll be okay." Um, so in any case, you can imagine what my enterotype might look like, where we have these different types of bacteria. So these bacteria that are changing, we have to go back and we'll take a look in the next diagram where these bacteria originally come from. There is a lot of controversy on this, even in the microbiome classes here at U of T. There's a lot of controversy as to whether or not in utero, when you're still in your mother's womb, whether or not your gut have bacteria in them or not. The majority, I would say about 75% of the individuals who study this, argue that you're born with a sterile gut from the time that you are born. So in utero, most people say that you don't have bacteria growing in your gut. Some individuals, some researchers have actually detected bacteria in neonates that are born. So as soon as they're born, they get swabbed, they take a sample of their esophagus, etc., and then they check to see if there are any bacteria, and they do find bacteria there. Very small proportion of individuals. And some individuals, when they check the amniotic fluid, the fluid surrounding the embryo in utero, they also find bacteria there. But again, a lot of controversy because some people say their samples are contaminated. If bacteria were growing there, why don't we have more issues with bacterial infection in younger neonates, etc. So there's a lot of controversy. I'm going to take the opinion that you're born without any bacteria at the time of birth. And one of the reasons why I think so, and this is the traditional data, which is the data that comes from the meconium. Those of you who have taken physiology previously, not maybe this semester, but have taken PSL 301 previously, do you know or recall what the meconium is? And I think it's on your crossword as well. So it is an important sort of term to go back and learn. Meconium? Lori, I think I know you know, but anyone else? Can you make a guess? Sorry? How do we measure bacteria, by the way, in your gut? We don't stick a probe up there. How do you measure bacteria? Oh, yes, I think someone swore, and that would probably be right. Yes, we measure it in your fecal matter, right? And so what's the first fecal matter that a newborn has? It's meconium, right? They don't really have their first poop, in other words, is known as this meconium. And when they measure this, and for those of you who have had the pleasures of changing the very first baby diaper, it's not a lot of fun. It looks like tar and it's gross and it's green and it's really terrible. And you kind of wonder if your baby is healthy or not. And I remember very clearly calling the public health department, telling them, my baby just pooped like this green tarry substance. I think she's really sick. And they told me to relax. It's fine. That's what's known as the meconium. So I thought I'd pass that along to you. This first poop that they have is free of viruses, and we don't see very many bacteria, which is one of the reasons why, because it's passing through the digestive system, why we think that they're not born with this intact gut microbiome. Again, we'll skip over this Hirschsprung's disease. We'll come back to things like the megacolon a little bit later on. Because we're a neurobiology class, that might be of some interest in coming back to this later. This is a really busy slide, and I'm putting this up here, and I'm going to be honest, I'm going to come back to this slide one more time in this course. But for the time being, the most important parts are the ones that I'm going to be showing you over here. As far as we know, the GI tract, the gastrointestinal tract of a newborn is sterile, no bacteria growing in it whatsoever. At the time of birth, whether you are born through a C-section or cesarean section, or you are born through vaginal delivery, can any of you notice a difference? Like, what's the big thing that is different for all of you when you look at this diagram? So if I'm born by a cesarean section or I'm born via vaginal delivery, do I have the same bacteria? Do I have the same enterotype? I do not. And so all of these bacteria, and again, I don't know what your microbiology backgrounds are like, but all of these bacteria are ones that we typically find on the skin, which makes sense that you're going to have more bacteria from the skin originally populating the GI tract of newborns that are born via the C-section. So things like Staphylococcus, Corynebacteria, etc. If you're born through vaginal delivery, you will have Lactobacillus, Prevotella, and Sneathia as the major bacterial enterotypes at the time that you are born. Okay, and again, that lactobacillus should be something that you have heard previously. It should be something that now you can understand why it colonizes the stomach, the intestine, and the colon in the way that I mentioned to you earlier. So again, I'm not going to go through all of these different things here. Between the time of 1 month to 6 months, we have a change in the bacteria that live within your gut. I don't need you to memorize that right now. I'm just telling you that your gut undergoes several changes in the gut microbiome as you get older. And by the time you are into this age, and for those of you who either have younger siblings or remember your younger siblings, what typically happens between 6 to 24 months? What do babies start to do then? And don't say walking or talking. What do they start to do? Yeah, they start to go on solid food. And when they start going on solid food, then we start going into the adult pattern, which is of the bacteroidetes as well as the firmicutes that we have here. And this pattern, after it's switched from the time that you were born into early childhood, and then by the time you get into adulthood, this pattern of bacteroidetes, the ratio of bacteroidetes to firmicutes, is going to be the one that you carry into adulthood. And if you're nice and healthy into adulthood, you will have the proper balance all the way through. But things like changes in your gut because of allergies, things where you've taken lots of antibiotics, things because you have changed the way that you eat, causing obesity or having an autoimmune disorder will change your gut microbiome. And again, the details of all of that, not so important. Okay, so you don't need to memorize all of the things that are here on the right. Any questions on this before we go on? So are you more excited by lunch now or less excited by lunch? Not sure. Are you guys eating lunch? So you definitely should go and eat lunch, but eat a nice healthy lunch. Yes.
Can you just repeat again what you said about the ratio of bacteroides to
So actually, I think that's coming up on another slide. So the ratio between the bacteroidetes and the firmicutes stay relatively stable if you're a healthy adult. So again, the virome seems to be a little bit different. From the best sort of knowledge that we have, it stays stable, but the virome doesn't change within the first 2 years of life. It seems to change within the first week of life. Most of the virome that you are born with changes over very, very quickly within that first year of life. Then it stays very, very stable throughout the rest of your life. So some of those early bacterial colonizers, like the lactobacilli, if you were wondering, they tend to be more aerobic. So they like having oxygen around, and these are likely coming from the maternal sources and coming through that vaginal delivery. And then as you start getting older, the bacteroids and the enterobacteria, which are the anaerobic types of bacteria, tend to dominate in the first year of life, and eventually by the time you reach your second year of life, when you're starting to eat more solid foods, you'll have the bacteroidetes and the firmicutes that tend to give you that stable ratio that lasts into adulthood. And then your gut microbiome stays that way for however long you're healthy until you reach old age. And then it all switches again. Okay? Was there a question? Yes.
I have a question about the different bacteria that you had from vaginal birth and
Yeah.
C-section. They
Yeah.
are different, but in the end, they still kind of end
That-
up going the same development.
That's right. So that's also been, yeah, you have it exactly right. So even though they start different, they do end up being very, very similar. And there's been a lot of controversy about that as well, Rachel, whether or not that early change or that early colonization by one type versus the other actually affects development, a lot of people are going back and looking, is there a correlation between the types of bacteria that you have through C-section and things like autism spectrum? So there is no correlation, but people were interested in trying to make those types of correlations. And let me just get through a couple of slides and then we'll try to wrap up just after 12 o'clock. So apologies, I'm not quite into the normal lecturing mode. I had sort of told you that we'll try to stick to about the 2-hour mark most weeks, and this week we'll do the same. If you have any questions, either about the course or about today's lecture, I'll be happy to stick around and answer any of those, or you can email me if you have anything that you've thought of later, or if you are listening in online later. So when we look at diet-induced models of obesity in mice or in rats, there tends to be this shift in that gut phyla. So we're not exactly the same as mice, and we see similar patterns that we do in mice. So go back to that Turnbaugh paper and the Wu paper. And overall, when you become obese, what happens is you tend to have a decrease in the levels or amounts of bacteroidetes, and you get an increase in the level of the firmicutes that occurs as you become more and more obese. Higher fat, higher sugar diets, you're going to get an increase in these phyla. Okay. And again, similar results in human twin studies show that there is a decrease in that bacteroidetes, but they don't see the same increase in firmicutes. They see an increase in humans of actinobacteria. Again, somewhat similar to the firmicutes that you see here, but they just found a different phyla of bacteria that tended to increase. So overall, the implication was, as you gain weight, as you become obese, as your diet changes from one that is healthy to one that is richer in fat as well as in sugar, you're changing your gut microbiome to have more and more of these firmicutes or the actinobacteria and less of these bacteroidetes. The problem is that the relative amounts of inflammatory cytokines that are being pumped out or being produced as a result of having these are different. Remember, some of these are gram-negative and some of these are gram-positive bacteria. So these result in very different inflammatory processes. Your gut becomes inflamed as a result of this change. Your brain doesn't take much longer to follow after it. So as you inflame your gut, you're also inflaming your brain as well. And again, all you need to do, at least according to this paper by Turnbaugh, both in 2006 and 2008, if I wanted to induce obesity, all I have to do is to spike your gut microbiome with someone who has obesity already, and it's enough to trigger obesity in you. Think how scary that is. But also think of the opposite. So if I can induce obesity in you by taking some of my gut microbiome and I gave it to you, and you were able to ingest it as a sort of sterilized pill, and it got into your gut microbiome, you would start to have the same bacteria in your gut that would produce obesity in you, at least according to Turnbaugh. But imagine the other scenario. Imagine if you are obese and you've been trying really hard to lose weight. You can't affect the way that you're taking in food for a variety of reasons that I'll get into in a moment. But imagine you took a nice healthy gut microbiome and established it in someone like me who needs to reduce the ones that are causing obesity. That might be a better way to start thinking about how to change the overall metabolic status. So not only can we induce obesity, but we might be able to induce fitness simply through a gut microbiome transplant. So this gets into the other wing. So how do you go from having a nice healthy diet? At one point, I can't really remember because it's been a really long time. At one point, I think I had a really healthy diet. At one point, I think I was eating relatively healthy food. But at some point, I became really interested in eating things that tasted really good. And how did all of that sort of occur? And how did that result in changes in my gut microbiome? Just like other things that cause reinforcement of behavior, we think that food itself could be a reward. Again, it was really, really controversial when this idea came out in the late 1990s, because it used to be thought only drugs of abuse are addictive and cause reinforcement behavior. Can't possibly be food. Um, but now we're relatively sure that you can have a food addiction that can be related to reinforced behaviors. So all of this goes back to basic neurobiology. So if you're looking for food, safety, being able to survive, etc., all of this is through reinforced behavior at the level of the midbrain. We have a small group of neurons in the midbrain, the mesolimbic and the mesocortical neurons, that actually allow you to engage in all of these self-sustaining behaviors. You need to be able to find safety, you need to be able to find food. When you get a reward from being able to find food or a safe place to stay, then you get a little reward out of it, and so it becomes a reinforcing type of behavior. So drugs of abuse, for example, like cocaine, override this. So normally cocaine doesn't exist in its purified state in nature, but we know that it can take over this particular pathway, and it can induce things that actually seem very much like the types of things that are up here. So if you've ever heard a drug addict talking about their need to take this drug that is being abused, they talk about how hungry they are for the taste of that particular drug, or taking that drug is better than sex, or they like the taste of that drug, or food tastes better when they're on that particular drug. All of that seems to suggest that the mesolimbic area is being reinforced by that. So I think what I might do is just leave you with this particular diagram. I know I have a few slides left, but maybe I'll come back to that in the next class and I'll kind of work that all in together. We're really talking about this one small circuit. So this circuit that I'm going to call the mesolimbic pathway, particularly the ventral tegmental area, or VTA neurons, that connect to the nucleus accumbens. This is sort of the reinforcing area in the brain related to rewards. If I think that that food is going to reward me in some way, or I take some pleasure out of that food, there's going to be more dopamine being released between the VTA and nucleus accumbens that causes me to have this reinforced behavior. I need that food eventually, or that particular type of food, not just to survive, because I like the feel of that food. As I said, how many of you are craving a food or have ever craved a food? Like maybe you're craving a really good Bueno chocolate bar. See, I keep going back to chocolate. So I know my brain is messed up, but if you're thinking about a craving, this is that pathway that is where you've reinforced the pleasurable aspects of that food that you need to go back and revisit. So we'll talk more about this mesolimbic pathway and how this change in behavior can cause a change in the gut microbiome, and as a result, how that might change your health status as well as your overall brain health as well in the next lecture. And I'll be happy to stick around if you have any questions. And for those of you at home, if you want to email me any questions that you have, please feel free to do so. Okay, have a great first half week back, and have a good lunch.